can't get here, or in case you want to revisit some of the things we'll talk about, I'll post the video once it's converted to an MP4, and then I'll get it on YouTube to make it easy to watch from wherever you are. So it looks like we're just about all here. I'm a little stuck for software right now. We're waiting for a couple of upgrades. We're waiting for a few upgrades from the university, actually for the university to pay for them, which would be really nice. We're a bit stuck here. This class is supposed to be done in the computer lab. And unfortunately, when they set up all the classrooms for social distancing, the one place that they did not set up for social distancing were the computer labs. So there's no partitions. Basically, everybody's on top of one another. If you've ever been in 245 or 345 in USC 1, everybody's kind of squished together. So I think we made the executive decision to say, okay, we still want to hold the course. I know everybody has trouble getting upper level bio courses when it comes to graduation. And not a lot of the courses have the kind of, I'll say, computational rigor that this one does. So I hope to get you really involved in doing some computer work. I realize that this is going to be a little bit of a challenge for some of you guys, because it's going to involve loading software on your home computer, laptop, whatever you're working with. If anybody is, I guess I should ask the question, is anybody working on campus or on a university computer? Oh, that's good, because it makes our life really easy. Good. Okay. Had somebody last semester working on a university computer and we weren't allowed to load any software on it. Brilliant move by the university. Brilliant stuff. So we're going to try to, so this will at least help us. So let me get started nice and easy with details everybody always wants to know. I'm just going to share my PowerPoint slides here, if I can find them. Don't panic. There we go. So hopefully everybody sees the PowerPoint stuff. And I'm just going to adjust. The one bad thing about this is I can't read the chat when I'm in this screen on my system here. So if anybody has a problem or thinks I'm not paying attention to their chats, that's got nothing to do with it. Just sing out, or I don't know, find some way to point me toward the chat, and we can get started. So basically, this is systems biology. I'm Guy Barbato. My official office is in USC 121. If you don't know where that is, it's on the first floor. If you enter by the water molecule, it's all the way down the hall on the left-hand side, tucked away in the corner. This semester, I will definitely be there on Tuesday, except today. Notice, not my office. Okay. If my truck, the water pump on my truck blew this morning, and so I'm stuck at home while they fix it. Needless to say, didn't make it to campus. Love old trucks. They're the joy of my existence. But at least starting next Tuesday, assuming I'm back on the road, I will be in Tuesdays and possibly one other day. It really just kind of depends on how things go with us. So I should be, at least if you need to see me in person, I will be around in the office on Tuesdays. My extension on campus is 4404, although I should warn you that I never answer the phone, especially because I'm not there. So they send me emails. The best way to get me is to email me. Real easy. You don't even have to remember how to spell my last name. I have a nickname from my account. It's just my initials, GFB, and the number 1 at stockton.edu. Easiest way to get me. I am usually available by email anytime, probably from 8 to about 11:30 at night or so. So I'm around. I still don't sleep very much. I think sleep is overrated. Also, in case you're interested, my Twitter handle is also at GFB and the number 1. I use Twitter, unlike past presidents, unlike a lot of crazy people online. I use Twitter only for communicating scientific information. And so it was pretty clear that even though I have a lot of students, I keep in contact with a lot of former students on Facebook. Facebook is not very conducive to sharing scientific information, and discovered that there are some really wonderful what I'll call ecological niches on Twitter, where you can really communicate with some top-notch scientists, and I share a lot of the things that I talk to folks about there. So anything about genetics, systems biology, usually nutrition. Some of you who've had me before may recall that my master's degree is in nutrition, and I postdoced at the Institute of Human Nutrition at Columbia Med School, where I worked on genetic models of obesity. So I do a lot of nutrition stuff. You pretty much name it, that's what I do. I'm not a lunatic tweeter. I'll tweet maybe once every day or two, really when something interesting happens. One of the ways that I'm going to manage this course is I try very hard to get the heck out of a Blackboard. I find Blackboard to be very cumbersome. It's clunky, it's kludgy, any kind of bad computer word you want to use, that pretty much describes Blackboard. So the only thing I really use Blackboard for is to send out bulk emails, and it does seem to do a pretty good job of that. But other than that, I'm really trying to stay away from it. For this particular course, I use a wiki program called Pbworks, and invitations have been sent to all of you. You should have received them. And it is the address is systemsbio.pbworks.com. All you have to do is accept the invitation. You are all writers on this wiki, which means you'll be able to create your own pages. You'll be able to write assignments. And basically, this is how we're going to communicate when we have assignments to do. And you could submit them right there. It works great. And when we talk about the assignments, and I'll show you this in just a second, really the grading for this, I know it's going to pain at least a few of you to learn this, but there are no exams. This is an upper level course. We don't do exams here. I'm assuming everybody's here because they want to work. There we go. I'm assuming everybody's here because they want to work and learn stuff. And as long as that's the case, we're not doing exams. However, the grading, unfortunately, we have to assign grades. The grades work like this. You get the weekly assignments. We will have weekly assignments, and you'll see they will be building in complexity as we go along. We'll start kind of simple and we'll work our way up. And then, of course, the other half will be the final project. Now, in the past, the final project, we actually had a poster session, and we took over the lobby of USC 1, and we had posters. We invited other faculty, other students, other classes, and it really was a very, very nice poster session. Of course, COVID screwed that up too. So what we're going to do is have an online poster session. There is a very real possibility that the NAMS symposium, the undergraduate research symposium for NAMS this year, will also be online. And if that's the case, maybe we'll submit some or all of our online posters there and use that as a way to communicate beyond the classroom. I think that decision hasn't been made yet. I think we should make that as a class a little bit later on. We'll decide on the final projects. Basically, the final project is you choosing an area of biology that you have interest in and creating a systems biology perspective to that project. And we'll talk about what that means over the next month, and probably in about a month, I'll have you guys choose your project, and we'll begin taking some of the computer work that we're talking about and applying it toward your project. And that's really going to, it's going to take us about a month or so to get there. And so just hold on for the ride a little bit. Before I switch over to the computer work and the assignment for this week, I thought it would be worthwhile for some of you who may be curious about this is what the heck is systems biology? I think that's a fair question. It is not as clear cut or as specific as you might imagine. And basically, it is a relatively new science. And what we're trying to do is integrate information at different levels to try to figure out how biological systems work and to see if we understand how information is integrated, if it can give us new insights into biology and biological systems. One of the great proponents of systems biology is a great biologist. He is started out as developing sequencing machines that currently we use many of his designs for both DNA and protein sequencing. He has some really unique inventions. He's at the University of Oregon right now, and Leroy Hood was one of the real proponents and originators of systems biology. And a more formal definition is that systems bio defines and analyzes interrelationships of all elements in a functioning system in order to understand how it works. Now, if you want to think about this in more physical terms, because some people are better at that, is think about this as creating a schematic diagram. For example, it could be a circuit board diagram. It could be a mechanical diagram of how your car works, how the water pump helps to cool your engine as you're driving along. It is that creating that kind of a schematic to illustrate how a biological system works. And as you'll see, we can use this to define almost any biological system. So it's got nothing to do really with the number of things in the system, or even necessarily the properties of the system individual elements, but rather what their relationships are. So if we think about what biology is really all about, biology is about mass, energy, and information and how they change through space and time. Okay, mass, energy, and information, and how those three things move through space and time. And they occur within specific networks. For example, when we think about mass and energy from a biological perspective, we're thinking about metabolism. Okay, how does the pizza you ate last night, okay, influence your digestive system? What gets absorbed into the blood? How are those nutrients dispersed through your body? How does it get broken down into its individual components so that those different molecules can be used by your cells? And then what do your cells do with them? What's the relationship among the different molecules? What are the products or byproducts that are formed? And ultimately, how does your physiology change as a result of that? So you see, this is a classic view of what a metabolic network is. The other way to think about it is the flow of information in DNA. And of course, this is the one we always think about. DNA as the classic, and we think about DNA in the context of how does the information in DNA flow through space and time? And the answer is we think about transcriptional regulation. How does DNA become transcribed and regulate gene expression? And also, what about the flow of information not involving DNA? And in this case, what we're really talking about is signaling. What kind of proteins or RNA signal expression of different proteins, control of different DNA elements? So we really have this whole idea of these individual networks. I want to give you some examples of what networks look like. This is a great one. This is air transportation networks. And if you notice, if you take a peek at this and just look at them, you'll notice a couple things. We got circles and little dots, and then we have lines among and between the dots. And you can figure out pretty quickly that the dots represent where the airport is located. So where in space are airports? Think the size of the dot has something to do with anything?
Maybe how busy or how large the area is?
Excellent. Or how large the area is, or maybe what within the context of
the volume.
Airline flights, right? The volume of what?
People passing through, amount of transportation going in and out.
Excellent, excellent. So the size of the dot indicates not so much the geographic area as it would on a map, but rather the amount of air traffic. How many flights? Perhaps how many people? They're probably correlated, right? You could actually overlay a couple of different things. We could actually create a dot, even though these circles are all colored with red. We might make a circle where the size of the circle or the diameter of the circle is related to the number of flights, and perhaps the color of the circle would indicate the number of people, or vice versa. So we can encode a lot of different information in a graphical unit. Obviously, the lines show connections between and amongst all the different networks. And in this case, the different colored lines actually represent different airlines. So just think about the amount of information that we've placed inside this simple graph. And what can we start thinking about? I mean, take a look at it. Does that help you understand issues in airports? Sure does to me. I personally love the ones going through Chicago there. Chicago's a big hub. I hope you have good air traffic controllers there. All right, there's a lot of traffic. Look at Chicago, Detroit, they're all smashed up all in there. Okay, so we can really use these kind of networks to understand some either underlying principles or perhaps even emergent principles that you might not predict if you worked at any given airport. Here's one I love. I love these. I think they're really pretty. This is actually an indication of, this is a mapping of the World Wide Web. It's actually a three-dimensional picture. I've got it here in 2 dimensions. I'll show you a little bit later on in the semester when we talk about how do you construct these networks. I'll show you some software that allows you to create this in 3 dimensions, which is really kind of cool. So you could rotate the picture and see how it grows. All of the different colors indicate the major servers and major hubs for the internet. The big one here in the middle, notice red is absolutely huge. You'll never guess who that is. Google. Okay, you'll notice the green in here, Microsoft. Okay, so we can look at all of the different connections and how they branch out. And since some of you have already taken some other biology courses, it looks frighteningly a little bit like a neural network. It looks like it's branching out neurons, and they're all branching out among one another and communicating with one another. So this is a pretty cool way to indicate. So in this case, we have each individual dot. We'll go to the middle here. I don't know if you could see my, wait a minute. Let's see if I can do this. Yeah, I guess I'll do black. Let's see. Hey, there we go. So we'll go to the main Google server here. Okay, so each individual dot in here is actually a server of one kind or another, and they are the number of other computers that connect to them. And you can actually see, for example, if we were to look how this little Google attaches to this other network, which is all in gray. And you could see how in each case, it spreads out to other networks. And of course, they end up branching out into smaller and smaller computers. All these tiny little, what you might want to consider dendrites, kind of if we think about a biological analogy, all these little computers out here are probably about the size of university or college networks. They are very, very far away, but ultimately connected to the major hubs. So really an interesting thing. There's an entire area of study in computational science of looking at how all of the different systems change in the World Wide Web to identify and predict traffic patterns, where more servers are going to be required, okay? How perhaps speed is going to be more necessary, increasing bandwidth. There are a lot of really important elements of the network that can only be investigated by looking at these three-dimensional graphic structures. I just love this picture. This is from this is actually an image of a social network. This is from data from Facebook. And again, this is each of the circles that you see are individual people that have lots of connections. And they were able to construct this with some really interesting software. This is far above some of the stuff we're going to do. But I just love the picture showing all the interconnections. And it really looks to me, boy, it just looks so biological to me, even though it has nothing to do with biology. I mean, except other than it's biological entities creating these social connections. But to me, it just looks like cells and interconnected cells. And but that's just me. It's also a cool picture. So what do we really mean when we talk about network? And a network, aside from being an intuitive feel of what the graph is, it's actually a mathematical structure, and it's a relatively new area of mathematics. And it's really anything where we're composing a series of points that are connected by lines. And network theory is actually an offshoot of graph theory. So when we think about graphs or networks, because we have points and lines, we talk about those points and lines as if they are nodes. So those circles are nodes that are connected by links. Okay, so you can see this has a very, very computational kind of vocabulary to it. So we're talking about nodes and links. And they're just some references. And these networks can be built for any functional system. Obviously, for our purposes, we're going to be talking about biological systems. So here's just some ways we could define graphs. They can be directed or undirected, meaning if we look at just 3 nodes, how many different ways can the 3 nodes be oriented? Well, they can be directed, and you could see, does the interaction, change my color here, does the interaction occur in one direction? Does it occur in both directions? And you could see there are all sorts of different types here. And of course, the other thing that we could do is we can quantitate these links by indicating the weight by the size of the line. We can weight them by, for example, the amount of information that might pass through these links. Okay, so they could be vertex weighted, meaning these are the weights referring to the size of the nodes, or they could be edge weighted, which is the information contained in the link itself. So again, these are just different ways to think about presenting network information.
Dr. Barbato,
Yeah,
what's the difference between directed and undirected?
directed just means a directional flow of information. So does it, for example, in this first one here, you could see that the links themselves are directed. They have direction.
So then just 6 is undirected?
Yeah, 6 would, sorry, I'm missing, let's see if I can, well, I can't move my pictures here. Yeah, 6 would be completely undirected.
Okay, thank you.
I'm missing one of the nodes on my pictures because my Zoom takes up a screen. Only got one monitor here, sorry. Sorry. So yeah, these are, and so these are just different ways to be thinking, because when you create your own network, for example, and we'll see examples of these, and we're not ready to do this for a while, but when you create it depends on what kind of an emphasis you want to give to your graphic. You may be interested in transfer of information, which means you may be more interested in the links and what's going on with the link, or you might be more interested in the size of each node. So you can have it, and in some very, very complex graphs, which are way off the charts, you can actually combine the approaches in almost any way. But that's really way more mathematically complex than we're going to deal with. But you ought to know that it happens. I mean, you ought to know that this is capable. Obviously, you can have all of your network can be connected. And again, you could see this is just a 6-member node, and they can be connected by all the different edges or by the links, or they can be completely disconnected. That is, in this one, for example, there are elements of the graph that are disconnected. Any of the networks that have no cycles are called trees. And by cycles, that means any individual loop. For example, here, you can create a circle. Okay, so if there's a circle, there's a cycle, meaning you go from one node and that you can follow the links all the way back to the original node. That's a cycle. If there are no cycles, then it's a tree. So you could think right away, what kind of graphic elements in biology have trees? Well, weirdly, phylogenetic trees, right? It encompasses the name. That's why it's called a tree. It's not called a tree because it looks like a biological branch or a tree branch. It's called a tree because there are no cycles. Now, that's not entirely true of all, for example, pedigrees. Sometimes they get a little more complicated, but we do make the assumption that there are no cycles. Alternatively, these cyclic graphs are wonderful for things like Krebs cycle, right? The citric acid or carboxylic acid cycle, okay? Any of these cycles, or if you want to think broader, that's kind of a metabolic network. What about an ecological network? The carbon cycle, the water cycle, okay? So we can use the same approach to look at molecules or to look at very large ecological constructs. And it's the same math, same pictures, same everything. So part of the reason for this introduction is I just want you to get you start thinking in these contexts, because whatever area of biology interests you most, and that's really what we're getting at here, you should be thinking to yourself, or you could be thinking, well, gee, how can I use this analysis if you're interested in diabetes in humans? Okay, you could possibly be thinking, well, I know there's cycles involved. So how could I fit insulin? How could I fit energy production, gluconeogenesis, glycolysis? How can I fit those concepts into a network graph? If you're interested in ecology, how can, for example, if I was to look at coastal ecologies, we know with global warming, seas are rising. If we construct a graph of coastal ecology, and then we postulate that water is going to rise, so a portion of that ecology is going to be either underwater or influenced by more water or warmer temperatures or whatever element you want, what would you predict would happen on the basis of your graph? So it's another way to approach a biological idea. These are just some different orientations. You could see, obviously, paths usually are directed. They go in one direction. We have star configurations. Here, of course, are our cycles, where you have little circles where you could start at any individual portion and on the way back to the, so for example, if these were labeled A, B, C, and D, you could start at A, work your way all the way back to A. These are cycles. Complete graphs very often have multiple connections among and between all of the different nodes. And this one's kind of fun down at the bottom. This is a bipartite graph, showing you relationships among and between different levels. Classic example of a bipartite graph might be our old buddy, Central Dogma. What if the green were DNA and the orange or red was RNA? You could see DNA can create different RNAs, perhaps those are different introns being spliced differently, and they can go back and interface or interact with one another. So that's kind of a classic way to look at a bipartite graph. So a lot of ways to apply these things to different biological concepts. When we think about where systems biology came from, and I like to go ahead and just show you this because it's really related to all of the different sequencing efforts that have occurred. When we think about molecular biology, we think about Central Dogma, for example. I mean, that is a classic view of a graph, right? You've got a little, when you think about Central Dogma, and I'll just draw it up here to make life easy, you've got DNA, right? And you got a graph, it reflexes on itself, RNA to proteins, I'll just draw P. And of course, now we know RNA can go back to DNA, so it's bidirectional. Okay, and we have all these different ways that we could present. So unknowingly, or at least unbeknownst to most people, in biology, we've been using network theory to describe biological concepts for really since the 1950s and 1960s. And ultimately, what's really great now is We have so much information from bioinformatics and from genomics to create these very, very large, complex models. We can look at things in very, very large sweeps, and we can really think in very broad terms instead of using the traditional reductionist mentality in science. And when I say reductionist, I mean cutting things into smaller and smaller pieces, right? That's the classic scientific method as devised by physicists and biologists, right? Physicists kept cutting matter up smaller and smaller, realizing that there were compounds, and compounds were made up of molecules, and molecules were made up of atoms, and atoms were made up of subatomic particles, and the subatomic particles even had smaller subatomic particles. Biologists had organisms, and then began to realize that there were cells, and then there were cell organelles, and then there were molecules involved in creating the cell organelles, and they had a function with them. So we always tended to think in science, this reductionist mentality, that we could parse things into smaller and smaller components, which would give us understanding of how the larger component worked. And to some degree, that's absolutely true. To some degree, we've made great progress like that. But does understanding DNA make you understand insulin and diabetes when you eat a slice of pizza for dinner the night before? So sometimes these complex systems have emergent properties of the system. They are more than the sum of their individual bits and pieces. And I'm sure you've heard that phrase before. Okay? So we really know enough now about the tiny little parts that we can begin to assemble them into larger systems and try to create new insights by using these techniques. So when we think again in terms of systems biology, and I've made this a little bit smaller and a little bit narrower, I'm not ignoring ecological models right now, but just to bring it to a little bit more local idea, because it's a little easier to think of in these terms, we can look at the different omic areas: genomics, transcriptomics, proteomics, metabolomics. Metabolomics, by the way, for me is one of the worst ever omics words ever invented. It's just crappy to say, and I'm not sure it's a really good description of what we talk about. But we can use annotation of these different systems to try to construct a biochemical network. We can even begin to create computer molecules. That's what we mean by in silico, meaning in silicon, like transistors. We can build computer models that could help us design experiments and eventually generate hypotheses that could help us understand the interactions of our original data here. And so that's the basic idea. There's some great examples here. Here's an example of the one that we've talked about. You could see we go from genome to messenger RNA regulation to in looking at the proteome and how the proteome interacts with genes, how the proteins interact with one another, and ultimately how these different proteins combine to create metabolism and the biochemical reactions. And of course, this is our buddy, the citric acid cycle in here. My graphic wasn't perfect, but it gives you an idea of how it does. So we can create these models all the way down. And we'll see some more examples of this as we go along. I love this one. This was from a paper, and it's already almost 2 decades out of time here. And these are all of the different proteins that have been isolated in Drosophila. These are all the different proteins isolated in Drosophila. We will see the software that actually developed this model, and you can actually break out into pieces, and you can actually break it out into even smaller pieces to identify each node is an individual protein that's been identified as being functional in Drosophila, and what other proteins they interact with. And you can see if this looks like a cell to you, that's exactly right. And things that occur in the green are nuclear proteins. Things that occur in the blue are cytoplasmic proteins. And things that occur in the yellow are extracellular proteins. So those are proteins that exist in the extracellular matrix. And if you were to look at that, you could already see there are many, many proteins involved in nuclear organization, which makes perfect sense in eukaryotes. It takes a lot of work to make a chromosome, to help chromosomes divide and keep the chromosomes in some form of chromosomal integrity. And then, of course, all of the metabolic issues are in the blue, all the proteins involved in metabolism. And then, of course, the extracellular matrix itself out there. So really kind of a fun thing. So we've really got these tremendous data sets we can do a lot of work with. This is a great chart. I'm not even going to try to blow this up. This is an example of all of the biochemical pathways in a eukaryotic cell. And if we were, and a little bit later on, these are put out just like maps. So you could look at individual areas, like we could look at F6 here. And in fact, if we looked at G5 to G6, let me get my eraser out because I'm going to get rid of this. Okay. You could see right away, guess what's dead in the middle? The most important cycle we've already mentioned a couple times, the citric acid cycle, right there in the middle, Krebs cycle, hugely important. And we can look at all of the interactions among and between all the different molecules in the networks. And actually, this comes from a great website. It's called Expazy. And we'll talk about Expazy a little bit later on. It's a great place to go if you want to look at metabolic networks or any kind of genomic or bioinformatic processes. It's part of the European Molecular Biology Group, and they have done a great job in presenting some of these different pathway diagrams. Really quite interesting. The Europeans, with the exception of Leroy Hood, the Europeans have always been much more interested in the sum total of networks and network diagrams than Americans. American scientists tend to be very reductionist. European scientists tend to be a little bit systems oriented. We'll look at this one again a little bit later on in the semester. This is one of my all-time favorites. This comes from a recent book on the biochemistry of toxins. And the title of the book is How Earth's Deadliest Creatures Mastered Biochemistry. And this is a great systems graphic of how all of these different species, and you can kind of see all the different species in their little graphic here, and where each of these species interrupts the clotting process in vertebrates. And so you can kind of see here's the clotting process, and then everything is labeled. For example, you could see here in number 2 in the green is these are kinins, and kinins are influenced by the venom in octopus, mosquitoes, certain amphibia, and of course bees. And so how all of these different creatures interrupt and disrupt the clotting process. So really kind of a cool way to look at it. I love these graphical images. They're just so powerful. We'll look at this in more detail and talk about the biochemistry a little bit later on. So I'm going to stop right here. I think it's a good stopping point. This is, of course, leading us into our next topic. Here is a great systems diagram. And you could see the guy's there. He's pointing at the diagram and he says, okay, that's why we need a computer. All right. So hopefully part of what I really did want to do here was to convince you, if you haven't been already, is to convince you that you really need to learn a little bit more about computers and computational systems in order to look at things from a systems perspective. All right. And so we're going to focus on that. I'm going to share one more screen, and then with any luck, this will work. Wait 1 second, please. I'm hoping this is it. All right, let's see if this works. Don't panic. Come on. There we go. Can you guys see the Pbworks page here?
Yes.
Okay, good. So this is just the front page of the Pbworks and all of the resources. This is a classic wiki. Okay, so this is a wiki, meaning that every page in here is related to a topic. And I've got a large amount of information here. The front page of the wiki has the assignments, which we'll look at in just a moment, but it also has texts and resources. In general, thinking in general, I really don't have a textbook for this course. There are a number of systems biology textbooks that are excellent. As you might imagine, they are very specific texts. They tend to be very expensive, and they tend to be very focused a little more deeply into computers and computation and programming than I really care to go in for our purposes. Now, we're going to inch our way into it, but we're going to this is kind of the equivalent of just testing out the water to see how cold it is. All right.
So if we've had no background on programming, we're all right.
Oh, yeah. Perfect. Then this is exactly where we want to be. This is exactly where we want to be. Yeah, this is, we'll get you to do some stuff, but won't make you go crazy. Won't make you go crazy. But I do have a variety of texts that are available, and you can see under this texts and resources area. There are some great resources here. I have gotten permission both from some colleagues, but also from a couple of publishers to share the information, share these books with you. If you like, you're welcome to download them onto your computer. And if you're interested in programming and you want to know more about it, there are a variety of books here. For example, we can look, for example, there's bioinformatics and Python, a lot of Python programming, also Perl. We'll talk about what these things mean in a bit, probably next week. But this really, all of these are available to you. You can either look at them. We'll use some sections of each of them throughout the semester. But if you'd like the entire book for yourself or for your library, they're all available as PDFs, and you can download them for yourself. More complicated in toto than what we're going to do here, but they should, but eventually you may find them useful. Also, we have our assignment page, which is located here, and I'm just going to switch to it. And you'll see that we've got a couple of assignments listed in here already. Our first assignment, which is really this week, is to get your computers up and running with software. Okay, and the software that we're going to be using Two types, we're going to be using R and we're going to be using Anaconda. And we'll talk about those in just a second. In fact, let me switch back here. There we go. There is a great primer here. You'll note the miscapitalization. And in this primer, come on, there are instructions on installing R on your computer. So the first step is to download and to install R on your computer. Now, the R website is very good at identifying the correct version for you to download and install when you get to the website. So for example, on my computer, if I say go to this website, there's a link, you'll see that it automatically correctly identifies me as working on a Windows machine. I happen to know mine is a 64-bit version of Windows, and all you have to do is download it. And again, I'm going to go back to, all you have to do is follow the directions. I've given you the simplest possible set of definitions to get it working. R is an open source program for statistical analysis. It is a phenomenal resource. It is a phenomenal resource. It's free. Excuse me. It's free, and I think every biologist ought to have a little bit of experience working with R. And so that's one of the reasons that we're going to use R. The second bit of software that I'm going to ask you to load, and again, we just got to there's some real simple instructions here. All you got to do is follow them along. There is also something called R Studio. So once you've successfully installed R, one of the things that you'll notice, and I'm just going to call up R on my computer here. Wait a sec. Ugh. Okay, it's not going to be friendly to me here. But the bottom line is it looks a little bit like this. If you run R straight up, you could see you get this thing. It's the R graphic user interface. See if I can expand this view a little bit, make it easier to see. There you go. And notice it's a console, which means that all you have to do is type commands. All right. We are probably never, ever going to use R as a console. I'm going to have you install R Studio, which will look something like this. And I'm going to just show you R Studio. One sec. Where'd you go, R? There you are. And R Studio looks something like this. And R Studio is great because if you look on the left hand, we've got our command line interface. And I'm just going to simply type in an equation. And notice it automatically gives me the answer. So R can behave as a simple calculator, okay? But the most important part of R are these two windows on the right-hand side of your screen, because these give you the ability to look for files, to actually generate plots, to keep track of your commands, so that let's say you would like to write a nice little program or have R do some statistics for you or create a graph and you want to save it. So these extra windows give you an opportunity to save your series of commands so that you can replicate exactly what you do. So we'll talk again. We're going to do examples of these a little bit, in fact, starting next week. But for the time being, I want to make sure that we can load R in RStudio. All right. The last thing, of course, there's one more, right? Is I want you to go ahead, and you'll notice in our assignment page, let me make this a little bit smaller here. So the first part is to follow those instructions to install R, and the link is right there to the primer, is to install something called Anaconda. Anaconda is a fantastic, brand new way to work with data. It allows you to do programming. It allows you to, Anaconda functions as a lab notebook, and it also encompasses RStudio. So what you're going to actually do is, if you poke down to the bottom, and you could read a little bit more about this, and there's a get started icon. And when you get started, what you're going to do is download Anaconda. And again, this works on all systems, Windows, Mac, and Linux. And in my case, because I have, even though my little laptop here is a couple of years old, I will do the 64-bit. It is a 64-bit processor. So I'd like you to give this a shot. I'm going to show you what Anaconda looks like. Oh, I have to stop my share, and I'm going to share the screen one more time. Here's what Anaconda looks like. And one of the great things about Anaconda is it's kind of a notebook that encompasses all of the different things that we're going to be able to use. Notice it encompasses RStudio. I don't think I can write on, no, I can't, shoot. Right in the middle here, we've got RStudio. We have Jupyter Lab, which is an electronic notebook. Here is another type of notebook, Jupyter Notebook. We have a couple of different data visualization programs. One of them is Orange. The other one is GlueViz over here. So we have these really powerful tools by just loading a single program. So Anaconda is really just a wonderful way. And I'll give you an introduction next week into Anaconda and how we're going to use it during the semester. If you're curious, you can actually, I'm going to show you the different environments. And we have all these different software programs available to us. Most importantly, we've got R, we've got RStudio. We also have Python installed as a result. So really kind of a fun, you'll see, it'll be fun. We have a really fun way to approach and get our computers up to speed. All the software I've chosen to this point functions on any computer. So it doesn't matter if you've got Windows, if you've got a Mac, or if you have a Linux computer, all of them work the same. They interact all the same. It'll get you all the software you need that we'll use throughout the rest of the course. So the job between here and Thursday is to try to load the software. I will give you a heads up, depending on how old your computer is, Anaconda can take a little while to load. All right? If you have any problems loading the software, just drop me an email or wait until Thursday, because we'll go over some of our experiences loading the software on Thursday. So either way is fine, depending on how anxious you are to get it up and running. The concept is to have it ready for next Tuesday, when we'll start looking at the properties and capabilities of these software programs in a little more detail. Okay? Cool.
Hey, Dr.
Panic
Barbata.
anybody into dropping?
No, I had a question about the assignments tab, Dr. Barbata.
Absolutely.
In the 126 state for the assignments due, there's a link for a Common Craft video that does not work.
Oh dear.
Is that necessary if we were able to get everything loaded?
No, if you got it loaded, it's not necessary.
All right. And then the one-page essay that's due on the
Nope,
4th.
nope. That's not yet. We haven't talked about that yet. Don't panic.
Okay.
Wait a minute. Let me find out where in the world I am here. Why is this being so cranky? Just give me 1 sec. Systems bio. There we go. Yeah, don't worry about that, because that's for next week. We'll talk about that next week. It's not due on the 4th. It's going to be assigned.
That's what my question was. Is it assigned on the 4th or is it due the 4th?
No. We got to make sure everybody's software is up and running. We're actually going to use R to make some word clouds. It'll be our first programming experience. It'll be fun. All right. Anything else? Going once? Twice? I don't know why my Zoom just got all goofy. There we go. Back. All right, give it a go. Have some fun. Remember, you can't break anything. If it doesn't work, no problem. We'll figure it out. That's the whole concept of this. It promises to be rather interesting, and I got to tell you, even working in the computer labs, whether it's 245 or 345, where our good buddies at IT down at the help desk actually load up all the software, we've had problems that we had to work our way through to get everything up and running. So don't panic. Okay, can't break anything, can't hurt anything, and hopefully, if you've got a problem, more than likely somebody else does as well. So like I said, drop me a quick email, or just wait till Thursday, up to you, depending on how busy you are and what things are going on.
One last
Okay?
question.
Absolutely.
Where are the readings located?
As they come about, we'll get them. Right now, we're following the primer to install R. They'll come
So
up
the
if each
readings are not due.
assignment comes up.
Okay.
All right. Have a good one. Stay dry. Looks like it's raining outside. Glad I piled my firewood this morning.
Thank you, Professor.
All right. We'll see you guys. Take care.
Take care.
Have a good one.